Okay, we're back to the book now. It's part 8. It's the urinary system. It's page 969. And we're over here on figure 2513. And it says here the transcellular and paracellular routes. And we're going through, here is the proximal convoluted tubule. Here, all of the different, it's a busy place, so I want you to sort of be aware of that. And here they're showing us the transport right here of we're inside the filtrate here. This is the filtrate that's going to become the urine. It hasn't become urine yet because we still have to take out the good parts and leave what is considered waste products alone. So here we have water. Water is passing through with its solutes. It's passing through this, how shall I say, aquaporin here, carrying solutes with it. And there's also, because it's carrying the solutes and the water is the solvent, then we describe this process as solvent drag, sort of a Scott Joplin sort of sensibility, if you will. But at any rate, we're getting into the blood with that. So these are the paratubular capillaries right here. And so this is what's going on with regard to that. There is various mechanisms for doing that. Over here, we're getting around a tight junction. Okay, so let me press on to the next page. Here we are. So, yeah. Here we are in the proximal convoluted tubule, page 970. You can see here's the loop of Henle and so on. And this is, they're showing us pretty much all the mechanisms here, and I'm sorry about all of the the messy annotations that I've made here, but I've tried to include as much as I possibly can. Again, we have solvent drag because we have water coming through its aquaporin. Who makes the aquaporin? It's ADH. Where does ADH come from? It comes from the posterior pituitary. Where is ADH actually synthesized? Is the hypothalamus. Slides down and gets deposited in the posterior pituitary. That's where it's stored. That's from where it is released. It becomes ADH. It acts right here. How do we make an aquaporin? Within a fraction of a second of your need. Okay, so it's a very subtle process. You're already sparing water. You might not be consciously thirsty because we have that problem with conscious override of everything. If we were our pet dog, the dog would be able to be aware of its need for water and would rehydrate within 1 gram of its need, right? So some animals have that all sort of configured into the whole thing. Here we have glucose, amino acids, vitamins, and so on. We're actually pumping that straight through. It's being pumped because there's this is a 2-seater right here. There's a seat right here for your whatever it is that you want. It could be the glucose, it could be the amino acid. There's another seat over here for the sodium. And so what we do is we have an ATP-driven sodium pump, pumps the sodium over here, and then we're going to be having potassium come out. So sodium goes from here over to here, and potassium comes from here to here. And then the sodium sits in this 2-seater. So this is called facilitated diffusion. Okay, so there's 2 kinds of facilitation going on here. There's the pump that is ATP driven, so this is an active transport, okay, sort of indirectly active. And there's no ATP directly here, but we've seeded a sodium here, and all this is waiting for is its transport molecule. So basically we're getting essential nutrients to sit here, and they automatically come through. Okay, so that's getting what we need into the blood, back into the blood. This is the same mechanisms that we would be using in the duodenum, even more so. This is much more sophisticated, actually, than the duodenum. Okay, so they do have in your book here little things that go from one spot, the proximal convoluted tubule, to the loop of Henle, and then we get on from there. We'll actually do a whole section on the loop of Henle. So that's part of what's coming up in the program next. They want to talk to us about antidiuretic hormone, but we've already just discussed that. And basically what we're doing with that is coming from the posterior pituitary, and we're coming down and we're making aquaporins when we need to spare water. We haven't had a drink of water in a while. The blood volume might drop down. We don't want anything to go into the urine that doesn't need to go into the urine, so we spare the water. Okay? And we have aldosterone. Aldosterone is going to go to the chief cells of the cortical collecting duct, and aldosterone is, as I indicate here, targeting the chief cells. They call it the principal cells here. Most physiologists say chief cells of the cortical collecting duct. Okay, and they forgot to put the word cortical there. Okay, and then and some parts of the distal convoluted tube. Now we know actually it's much more the cortical collecting duct. This is a slightly older edition, but the idea here is that we're going to be pumping sodium. Okay, so we come to this big overview here, this diagram. Here we are. What's the legend say here in terms of color coding and so on? The legend says this right here. The blue is for reabsorption and the green is for secretion. Let's take a look. We have our filtrate, and the blue says that we're going to get back all of this stuff here in the proximal convoluted tubule. We're going to get back 65% of the filtrate. We're going to get the water, the sodium, bicarb. We're going to get glucose, other essential, that means to say essential nutrients there. That's the blue box. Here is the green box. We're going to get rid of some stuff that we don't want. We don't want extra hydrogen ion. Okay, we don't want ammonia, basically, and urea. If we happen to be using a drug protocol, usually it's the drugs that get secreted here. Remember, this is reabsorption. When it comes this way, it's it's filtration. So reabsorption, filtration, reabsorption. That's the main thing that's going on. ATP driven, also ATP driven is the secretion. So I'm not getting rid of enough hydrogen ion. So not only will I filter it and leave it there to become urine, but I'm going to actually pump it right here, which is called secretion. Okay, and the same thing with the naughty drugs that we don't want. The same thing with the anything related to the amino acid that needs to come, the amino group from the amino acids. So urea and other byproducts related to urea, uric acid, so on. Then we come to the descending tubule here of Henle's loop. And what's unique and special about this is that water can leave, but sodium can neither enter nor leave. It's impervious to sodium. Okay? And so what's happening here is notice how the color here is getting darker, darker, darker very gradually. The artist is trying to denote the idea that we're having an increased concentration as the sodium increases. So what's going on here is more and more water is going to come out, all right, because it's more and more tonic out here. And that's going to increase the tonicity of the filtrate. So the filtrate, as the water comes out, gets ever more concentrated and sludgy, okay, just the way you like it. Then once we get to here, there's no longer any pathway for the water, and now only sodium can leave. Okay, so at first it leaves passively. Here we have this little reabsorption number here. Okay, and then once we get to the thick portion here, we're going to get pumping, and we're going to pump out sodium and even chloride. Okay, lots of pumping going on here. Okay, and water can't move here. Water is all done, and we're taking that really concentrated soon-to-be urine, and we're taking all the sodium out to increase our tonicity down here. Okay, and then we come up this way. Once we get to here, we're actually at the macula densa cells. That's at the very end of the thick loop of the Henle right here because it's still doing a little bit of sodium pumping. Okay, and then we get out to our distal convoluted tubule for fine tuning, and we'll pick it up right there in the next part.